Botulinum toxin is a neurotoxin primarily produced by Clostridium botulinum, a bacterium known to cause food poisoning. There are a total of 8 subtypes of the toxin, with different onset and duration of action. Currently, only types A and B are commercially available. The structure of the toxin varies from a simple protein structure that causes the toxic effect and a more complex structure that involves proteins that protect these structures. Among them, the 900 kilodalton structure consists of the neurotoxin structure responsible for toxic effects, and the non-toxic non-hemagglutinin and hemagglutinin structures, which surrounds and stabilizes the neurotoxin. Generally, for a muscle to contract, an electrical signal must be transmitted from the brain to the muscle via nerves. This signal transmission between cells is mediated by neurotransmitters, such as acetylcholine. The electrical signals transmitted through nerve cells induce the secretion of neurotransmitter, and the transmitters bind to the receptors on the muscle cells, causing muscle contraction. Botulinum toxin prevents electrical signal transmission by inhibiting neurotransmitter secretion and thereby prevents muscle contraction. This mechanism also works on facial muscles that create various facial expressions. When administered into the facial muscles, the toxin interferes with the facial muscle contraction, thereby improving facial wrinkles caused by muscle contractions. The name Nabota comes from natural botulinum toxin type A. It is a botulinum toxin type A product with the generic name probotulinum toxin A. Nabota is available as 4 dosage units: 50, 100, 150, and 200 units. As of 2023, Nabota has gained approval for 5 indications, including glabellar lines, lateral canthal lines, post-stroke upper limb spasticity, and benign essential blepharospasm, and most importantly, benign masseteric hypertrophy. Nabota received the world's first indication approval for benign masseteric hypertrophy as a botulinum toxin, making it the first neurotoxin product to be approved for both upper and lower face indications. Nabota, which was first approved by the Korean Regulatory Agency, MFDS, in 2013, has been approved by 3 major global regulatory agencies: Health Canada, US FDA, and EU EMA in 2018 and 2019. This makes Nabota the first Asian botulinum toxin product to be approved by 3 major regulatory agencies, including US FDA. And in 2024, Nabota celebrates its 10th anniversary for first product launch in Korea. Nabota has been actively expanding its business worldwide, and as of December 2023, Nabota gained approval in 67 countries, partnered with over 80 countries, and marketed in 30 countries, strengthening its position as a global botulinum toxin product. Nabota undergoes a series of processes, including cultivation, precipitation, purification, vial filling, and vacuum drying. High Pure technology is a globally patented technology developed by Daewoo Pharmaceutical that involves the precipitation and purification processes, and that allows Nabota to be produced as a high-purity toxin. First, cells are extracted from the cell bank and cultivated. The impurities formed during the cultivation process is subsequently removed through the precipitation process. During the purification process, more impurities and neurotoxin molecules other than 900 kilodalton are removed through 2 rounds of anion exchange chromatography, resulting in a high-purity toxin with over 98% of 900 kilodalton toxin. The purified, high-purity toxin is filled into each vial to undergo drying process and afterwards finally packaged as a product. Unlike many biologics produced through freeze-drying, Nabota adopted vacuum drying method. The freezing process during the freeze-drying method may lead to formation of ice nuclei, which could increase the risk of inactive toxin formation. Due to this feature of freeze-drying process, it may be less suitable for botulinum toxin products with a minuscule amount of active ingredients. Nabota, by adopting vacuum drying method, which does not involve the freezing process, reduced the risk of inactive toxin formation. Currently, all Nabota products, excluding the 150-unit product, are produced through the vacuum drying method. Nabota is the first Asian botulinum toxin approved by the top 3 major global regulatory agencies, including US FDA. Nabota is a high-purity 900 kilodalton toxin produced through patented technology and vacuum drying. Nabota is produced as a high-purity toxin through Daewoo Pharmaceuticals' globally patented high-pure technology. The purity test results confirm Nabota's high purity, consisting of over 98% of the 900 kilodalton complex. Additionally, it is produced using the vacuum drying method that does not involve a freezing process, thereby minimizing the risk of inactive toxin formation. Nabota is a predictable toxin with rapid onset of action and targeted effect. Nabota demonstrated efficacy in 85.4% of all test subjects as early as the second day of treatment, proving its rapid onset for improving glabellar lines. The onset of action significantly influences patients' satisfaction with their cosmetic treatments and their product selection, as they often prefer products with rapid onset of action. Furthermore, to achieve predictable and natural results through botulinum toxin treatments, it is essential that the injected toxin works on the targeted treatment area with precise diffusion and targeted effect. Nabota has been proven to diffuse more precisely from the injection site compared to other products, resulting in targeted effects on the treatment area. The precise diffusion of Nabota may minimize diffusion to other unwanted areas and deliver predictable results. Finally, Nabota is toxin with proven immunogenic safety in large-scale clinical studies. Botulinum toxin consists of neurotoxin and a complex protein. Antibodies specifically formed against the neurotoxin, which produces the toxic effect, are known as neutralizing antibodies. Neutralizing antibodies alter the efficacy of the toxin and serve as a factor in inducing resistance. Therefore, confirming immunogenic safety of a toxin is crucial. Nabota has proven its immunogenic safety under single-dose, repeat-dose administration, and in administrations of a maximum of 360 units in over 1,500 subjects through large-scale clinical studies. No neutralizing antibodies were newly formed during global and Korean single-dose glabellar line studies, as well as in repeat-dose studies, and in post-stroke upper limb spasticity study with a maximum of 360 units administered. Nabota has conducted large-scale clinical studies on glabellar line improvement in the United States, Europe, and Canada with over 2,000 subjects. Through 2 Phase 3 clinical studies, EV001 and EV002, Nabota proved its efficacy on glabellar line improvement compared to placebo with 67.5% and 70.4% of the subjects showing at least 2-point reduction on GLS score. The EV003 study, a head-to-head comparative study with Allergan's Botox, proved Nabota's non-inferiority against the competing product. Furthermore, Nabota-treated groups showed a tendency for higher responder rates for positive responses on the GAIAS, Global Aesthetic Improvement Scale, compared to the competing product for both subjects and investigators. Lastly, Nabota's efficacy and safety were proven in 2 repeat dose studies, EV004 and EV006. Over 90% of subjects achieved and maintained their glabellar line improvement of at least 1-point reduction in GLS score, even after up to 4 treatments over a year. Moreover, no neutralizing antibodies formed, and no serious adverse events were reported in this study, confirming Nabota's safety in repeat dose administrations. As interest towards neurotoxin treatments are growing in all populations, regardless of age, gender, and skin color, post-hoc analyses studies were conducted. These studies, based on the EV001, EV002, and EV003 data, were conducted to confirm its efficacy and safety for glabellar line improvement in millennials, males, patients with skin of color, and patients aged 65 years old or older. In addition, Nabota proved its efficacy and safety with extra strength administration of 40 units, which doubled the dose to 40 units and concentration to 8 units point, and thereby offering a new treatment option. Among the 3 groups, only the prabotulinum 40-unit group showed that more than 50% responder rate, 1-point reduction of GLS score at up to 26 weeks or 6 months, proving its long-term effects for glabellar lines improvement. An important factor that determines facial contour is masseter muscle hypertrophy. Nabota proved its significant efficacy and safety in improving masseter muscle hypertrophy among Koreans through a Phase 3 clinical study, and became the world's first botulinum toxin to gain approval for masseter muscle hypertrophy. Additionally, the open-label extension study proved that subjects who received Nabota showed a greater improvement after the second round of administration compared to the first round. Products that have undergone a drying process may have different physical formation depending on the drying process. Vacuum-dried products, like Nabota, appear as white or nearly transparent, resembling an unstructured cake, and the vial may seem empty. However, this characteristic results from the vacuum drying process and does not affect the quality of the product. When diluting Nabota, the dilution ratio is generally 4 units per 0.1 milliliter for facial procedures and 2 units per 0.1 milliliter for body procedures, but this dilution ratio can be adjusted. Open the Nabota box. Remove the cap after taking out the vial. Wipe the rubber stopper with alcohol or ethanol gauze and let it dry completely. Open the normal saline. Inject the syringe to the normal saline and extract the solution. Insert the syringe containing the extracted saline into the rubber stopper of Nabota's vial. Note that to prevent coring, position the needle bevel upwards and approach the vial surface at an angle of 45 to 60 degrees. Apply downward pressure on the needle while gradually bringing it up to an upright position until it completely penetrates the rubber stopper. As soon as the needle fully penetrates the rubber stopper, tilt the syringe to touch the vial wall and slowly inject the diluent along the wall. Remove the needle from the vial and gently roll the vial between your palms to dilute Nabota with the saline solution. During this process, avoid shaking too vigorously to prevent bubbles from forming in the vial. Gently turn the vial upside down to ensure that the diluent reaches the rubber cap at the top of the vial. Likewise, be cautious not to shake vigorously to prevent bubble formation. Leave the vial for 60 seconds. As interest in hair loss grows, botulinum toxin is being suggested as a treatment option. Nabota 100 units are diluted to a final concentration of 2 units per 0.1 milliliter and administered by intrafollicular injection into the hair follicle area where dermal papilla cells exist. Approximately 20 to 30 injection points are set at intervals of 1 to 2 centimeter on the hair loss area, and 2 units are injected into each point at a depth of 4 millimeter. A total of 50 units are administered per treatment session, and the treatment can be conducted monthly, with a total of 6 treatments over 6 months. Nabota has conducted clinical studies using differentiated treatment method. In these studies, when Nabota was administered intrafollicularly to hair loss patients, the number of hairs significantly increased after 24 weeks following injection compared to baseline, confirming the efficacy of Nabota for treating hair loss. Additionally, various case studies are being conducted to confirm the effectiveness of Nabota combined with other treatments. Body toxin procedures are gaining popularity among Korean women in their 20s and 30s, mainly for shoulder and calf contouring. For shoulder contouring, Nabota 200 units are diluted in 4 milliliter of normal saline and injected in the upper trapezius muscle at areas B and C, where the nerve terminals are concentrated. For each side of the trapezius, 6 injection sites are set at 2 centimeter interval, and 5 units are injected at each injection point. For leg contouring, Nabota 200 units are diluted in 4 milliliter of normal saline, and 10 units are injected into each of 6 injection sites on the medial and 4 sites on the lateral gastrocnemius muscle. The results of 7 clinical studies using Nabota have been published for body toxin, each proving the efficacy and safety of Nabota on trapezius, calf, and deltoid muscle contouring. NaboLift is an intradermal injection treatment method, while commonly referred to as dermatoxin or skin Botox. NaboLift is a collection of unique administration regimen on different injection sites designed for Nabota by Nabota's specialist doctors. ID injection can achieve effects such as skin tone improvement, reduction in pore size, and lifting through various mechanisms caused by injection into the skin surface layer. Different doses are administered to each part of the face, with 0.5 to 2 units injected per site. Hyperhidrosis refers to a chronic dysfunction of the autonomic nervous system that causes abnormally excessive sweating in amounts greater than needed to control body temperature without a particular inducer. Before the procedure, apply the anesthetic cream to the injection site, wrap the area, and wipe it off. Aerosol spray can also be used as a time-saving protocol. Dilute 100 units of Nabota in 4 milliliter of normal saline to make a final concentration of 2.5 units per 0.1 milliliter on excessive sweating area in the axilla. 0.1 to 0.2 milliliter each is evenly injected intradermally into 10 to 15 injection sites at an interval of approximately 1 to 2 centimeter. A total of 50 units are injected for each side. Through a series of chapters, we have introduced Nabota and differentiated treatment methods. Nabota takes pride as Asia's first U.S. FDA-approved botulinum toxin and will continue to grow as a leading product in the global toxin market.